Hello there, and welcome to the Sleepy Science Channel. Tonight, we begin a gentle journey into one of the most fundamental rhythms of existence itself. A rhythm that pulses quietly within you at this very moment, orchestrating the rise and fall of hormones, the ebb and flow of body temperature, and the gentle transitions between wakefulness and sleep. This is the story of your circadian rhythm, your body's internal clock that marks time not in seconds or minutes, but in the ancient language of light and darkness. It is a rhythm as old as life itself, carved into your cells over millions of years of evolution, a timekeeper that connects you to the rotation of the earth and the daily dance between day and night. Tonight, we will explore how this biological clock works, nestled deep within the brain in a tiny cluster of cells no larger than a grain of rice, yet powerful enough to influence every system in your body. We will discover the molecular gears that turn within each cell, the hormones that rise and fall like tides, and the genetic variations that make some of us morning people while others come alive after dark. We will travel through time, from the first observations of plants opening their leaves to the sun, to the modern laboratories where scientists peer into the inner workings of clock genes. We will examine what happens when this rhythm is disrupted, when we cross time zones or work through the night, forcing our ancient clock to adapt to a modern world that never sleeps. If you enjoy these videos and would like to join us for more nighttime explorations, you can like and subscribe below, and feel free to share your thoughts down in the comments. But for now, all you need to do is get comfortable. Let your breathing slow, your mind soften, and allow yourself to drift into the gentle rhythm of time itself. Let's begin. The discovery of biological time. Long before scientists understood the intricate machinery of the circadian clock, humans observed something curious in the natural world. Plants seemed to know when morning would come. They would open their leaves to greet the sun, close them at night, all without any apparent signal from the environment. This phenomenon puzzled naturalists for centuries. In 1729, a French astronomer named Jean-Jacques d'Autus de Méran conducted a simple yet profound experiment. He took a heliotrope plant, one that famously opened its leaves during the day and closed them at night, and placed it in complete darkness. The expectation was clear. Without sunlight to guide it, the plant would remain confused, its leaves neither opening nor closing in any predictable pattern. But that is not what happened. To de Méran's astonishment, the plant continued its daily cycle. Even in total darkness, the leaves opened in the morning hours and closed in the evening, as if the plant carried within itself some internal knowledge of time. This observation marked one of the first documented encounters with what we now call an endogenous rhythm, a biological cycle that persists even without external cues. For decades, this observation remained more curiosity than science. Naturalists and early biologists noted similar patterns in animals. Bees would return to feeding sites at specific times of day. Birds would migrate according to seasonal changes. Small mammals showed predictable patterns of activity and rest. But the mechanism behind these behaviors remained shrouded in mystery. The early 20th century brought renewed scientific interest in biological rhythms. A German botanist named Erwin Bunning began conducting meticulous experiments with bean plants in the 1920s and 30s. He demonstrated that the period of these rhythms, the length of one complete cycle, was inherited, passed from parent plants to their offspring through genetic material. This suggested that biological clocks were not learned behaviors or responses to subtle environmental cues that experimenters had missed, but rather were coded into the very fabric of life itself. Bunning's work laid crucial groundwork, but it was the research that followed in the 1940s and 50s that would truly revolutionize our understanding. At the University of Minnesota, a young physician-scientist named Franz Hallberg was studying the adrenal glands in mice. He noticed something peculiar. The size and activity of these glands changed systematically throughout the day. The mice showed clear rhythmic patterns in their hormone production, patterns that persisted even when the animals were kept in constant conditions. Hallberg was meticulous in his observations. He noted that these rhythms were not exactly 24 hours long. Instead, they approximated 24 hours, running slightly longer or shorter depending on the individual and the conditions. He needed a term to describe these near 24-hour rhythms, something that would capture their approximate daily nature, without implying they were exactly locked to the rotation of the Earth. In 1959, Franz Hallberg introduced a term that would define an entire field of study. He combined two Latin words, circa, meaning about or approximately, and diem, meaning day. Thus was born the word circadian, describing rhythms that cycle approximately once per day. This seemingly simple act of naming would prove transformative, giving scientists a precise vocabulary to discuss these biological phenomena and helping to establish chronobiology as a legitimate scientific discipline. But Hallberg did more than name these rhythms. Through his careful experiments, he demonstrated that circadian rhythms were not mere responses to environmental cycles, but were generated internally by the organism itself. He showed that these rhythms could be shifted and synchronized by external time cues, which he called Zeitgebers, a German word meaning time givers. The most powerful Zeitgeber he found was the cycle of light and darkness. Around the same time, another crucial piece of the puzzle was falling into place. Colin Pittendry, working at Princeton University, was studying fruit flies. He observed that the process by which these flies emerged from their pupal cases, a behavior called eclosion, occurred in waves at specific times of day. When he maintained the flies in constant darkness, the rhythm of eclosion continued, proving it was driven by an internal clock rather than by subtle environmental cues. Pittendry's experiments were elegant in their precision. He demonstrated that while temperature affected many aspects of the fly's biology, it did not significantly alter the period of their circadian rhythm. This temperature compensation, as it came to be known, was a hallmark of true circadian clocks. Researchers have proposed that a clock that sped up or slowed down dramatically with temperature changes would be useless as a timekeeper in the variable conditions of the natural world. These discoveries of the 1950s established several fundamental properties of circadian rhythms. First, they persist in constant conditions, proving they are endogenously generated. Second, they can be entrained or synchronized by environmental cues, particularly light. Third, they are temperature compensated, maintaining their period across a range of temperatures. And fourth, they are genetically determined, varying in their characteristics from individual to individual and species to species. The work of these pioneers opened the door to decades of research that would follow. Scientists began searching for the anatomical location of the biological clock. Where in the body did this mysterious timekeeper reside? In mammals, the answer would eventually point to a tiny region in the brain, but that discovery would require years of painstaking investigation. The master clock, the suprachiasmatic nucleus. In 1972, two independent research teams made a discovery that would fundamentally transform our understanding of circadian rhythms in mammals. Robert Moore and Victor Eichler, working at the University of Chicago, and Frederick Stephan and Irving Zucker at the University of California, Berkeley, published studies showing that lesions to a specific tiny region of the brain abolished circadian rhythms entirely. The region they identified was the suprachiasmatic nucleus, often abbreviated as SCN, a pair of small clusters of neurons in the hypothalamus. When these nuclei were destroyed, laboratory animals lost their normal patterns of activity and rest. They would eat, drink, and sleep at random times of day, with no discernible rhythm. Their body temperature no longer followed its usual daily pattern. The secretion of hormones that normally showed clear circadian rhythms became arrhythmic. But when the suprachiasmatic nucleus remained intact, these rhythms persisted even in constant darkness or constant light, demonstrating that the SCN was not simply responding to environmental cues, but generating rhythms from within. Further confirmation came from transplant experiments. When fetal suprachiasmatic tissue was transplanted into animals whose own SCN had been destroyed, circadian rhythms were restored. The transplanted tissue imposed its own characteristic rhythm on the host animal, proving that the SCN was indeed the source of circadian timekeeping. The suprachiasmatic nucleus is remarkably small, given its importance. In humans, each nucleus contains only about 10 to 20,000 neurons, a tiny fraction of the roughly 86 billion neurons in the entire brain. If you could look at the SCN in cross-section, you would see a structure roughly oval or almond-shaped, measuring only about 0.5 millimeter across, truly no larger than a grain of rice. The location of the suprachiasmatic nucleus is not accidental. Its position directly above the optic chiasm, the point where the optic nerves intersect, places it in the perfect location to receive information about light from the eyes. This anatomical arrangement reflects the nucleus's primary function, to synchronize the body's internal rhythms with the external cycle of day and night. The suprachiasmatic nucleus is bilateral, meaning you have two of them, one on each side of the brain, symmetrically positioned along the midline. Each nucleus is roughly oval in shape, though some researchers have noted they appear more elongated in certain individuals. Despite their small size, these nuclei contain a remarkably organized structure, with different types of neurons arranged in distinct subregions, each playing specialized roles in the generation and distribution of circadian signals. Within the suprachiasmatic nucleus, researchers have identified two main anatomical divisions, the core and the shell. These regions differ not only in their location, but also in the types of neurons they contain, the neurotransmitters these neurons produce, and the functions they serve in the larger circadian system. The core of the suprachiasmatic nucleus, located in the ventral or lower portion of the structure, is the first to receive light information from the eyes. Neurons in the core produce two primary neurotransmitters: vasoactive intestinal polypeptide, abbreviated VIP, and gastrin-releasing peptide, abbreviated GRP. These chemical messengers play crucial roles in synchronizing the individual neurons of the SCN with each other and in responding to light signals from the environment. The shell of the suprachiasmatic nucleus, positioned dorsally or above the core, contains neurons that produce arginine vasopressin, abbreviated AVP. These neurons send signals to other parts of the brain and body, translating the timekeeping functions of the SCN into coordinated rhythms of physiology and behavior. The shell neurons are responsible for much of the output from the suprachiasmatic nucleus, communicating time-of-day information to regulate sleep and wakefulness, hormone secretion, body temperature, and countless other processes. The connection between the eyes and the suprachiasmatic nucleus is mediated by a specialized pathway called the retinohypothalamic tract. This neural pathway originates not from the familiar rod and cone cells that enable you to see images and colors, but from a distinct population of photosensitive retinal ganglion cells. These specialized cells contain a photopigment called melanopsin, which is particularly sensitive to blue wavelengths of light, with peak sensitivity around 480 nanometers. When light enters your eyes, these melanopsin-containing cells send signals directly to the suprachiasmatic nucleus via the retinohypothalamic tract. The neurotransmitter they use is glutamate, and its release onto SCN neurons triggers a cascade of molecular events that can shift the phase of the circadian clock. This is how light, the most powerful Zeitgeber, synchronizes your internal clock to the external world. But light is not the only influence on the suprachiasmatic nucleus. A second neural pathway, called the genicular-hypothalamic tract, provides indirect input from the eyes via a region called the intergeniculate leaflet. This pathway uses the neurotransmitter neuropeptide Y and appears to integrate information about both light and behavioral activity. Based on what we understand, it may play a role in how non-photic cues, such as exercise or social interactions, can influence the circadian clock. Within the suprachiasmatic nucleus itself, individual neurons are remarkable timekeepers. When isolated in a laboratory dish, separated from all other cells and from any environmental signals, a single SCN neuron will continue to show rhythmic activity. Its membrane potential, the electrical charge across its cell membrane, will oscillate in a roughly 24-hour pattern. The rate at which it fires action potentials will rise during what would be daytime and fall during what would be night. This cellular autonomy is extraordinary. It means that each neuron in the suprachiasmatic nucleus contains within itself the molecular machinery necessary to generate circadian rhythms. The neuron does not need external signals to tell it what time it is. Instead, it generates time from within, through the rhythmic expression of clock genes and the accumulation and degradation of their protein products. However, while individual neurons can keep time on their own, the power of the suprachiasmatic nucleus comes from the coordination of these 20,000 individual cellular clocks into a unified whole. This coordination requires communication between neurons, and the neurotransmitters VYP and AVP play essential roles in this process. Research has shown that VIP signaling through its receptor, called VPAC2, is absolutely critical for maintaining synchronized rhythms within the suprachiasmatic nucleus. Animals lacking functional VIP or PAC2 receptors show disrupted circadian rhythms because their SCN neurons fail to coordinate with each other. Each neuron continues to oscillate, but they do so at slightly different periods and phases, producing a cacophony rather than a symphony. The molecular mechanisms within SCN neurons involve intricate feedback loops of gene expression. During the subjective day, genes called clock and BMAL1 are active, producing proteins that form complexes and bind to specific regions of DNA. These protein complexes then activate the transcription of period and cryptochrome genes. As period and cryptochrome proteins accumulate in the cell, they eventually form their own complexes that move back into the cell nucleus and inhibit the very clock and BMAL1 proteins that initiated their production. This negative feedback loop is the core mechanism of the cellular clock. It takes approximately 24 hours for one complete cycle, for period and cryptochrome proteins to be produced, to accumulate, to move into the nucleus, to inhibit clock and BMAL1, to be degraded, and for the cycle to begin again. Additional layers of regulation involving phosphorylation of proteins by kinases and their subsequent degradation add precision and stability to this fundamental oscillation. The electrical activity of suprachiasmatic nucleus neurons follows this molecular rhythm. During the day, when core clock gene expression reaches certain phases, neurons become more excitable. They depolarize, meaning their membrane potential becomes less negative, and they fire action potentials more frequently. This daytime firing can reach rates of 10 to 15 action potentials per second or more. At night, the neurons become hyperpolarized, more electrically negative, and their firing rate drops dramatically, sometimes to near silence. This daily oscillation in electrical activity is driven by rhythmic changes in ion channels, the proteins that control the flow of charged particles across the cell membrane, potassium channels, which allow positive potassium ions to leave the cell, show circadian variation in their expression and function. During the night, increased potassium conductance helps keep neurons hyperpolarized and quiet. During the day, reduced potassium conductance and increased sodium conductance through channels like NALCN promote depolarization and active firing. The rhythmic output from the suprachiasmatic nucleus influences virtually every aspect of physiology. The SCN sends direct neural projections to other regions of the hypothalamus, including the dorsomedial hypothalamic nucleus, which coordinates circadian rhythms of feeding and activity, and the paraventricular nucleus, which regulates the release of hormones from the pituitary gland and controls the sympathetic nervous system. One of the most important outputs from the suprachiasmatic nucleus is its control over melatonin production by the pineal gland. This control is not direct, but rather operates through a multisynaptic pathway. SCN neurons project to the paraventricular nucleus, which in turn projects to neurons in the spinal cord, which then project to the superior cervical ganglion. From there, sympathetic nerve fibers finally reach the pineal gland, where the release of norepinephrine triggers melatonin synthesis. This elaborate pathway ensures that melatonin is produced primarily at night, when the suprachiasmatic nucleus is electrically quiet. During the day, when SCN neurons are highly active, they inhibit this pathway, preventing melatonin synthesis. The result is a robust circadian rhythm of melatonin that serves as an internal signal of darkness, helping to coordinate sleep and other nighttime processes. The suprachiasmatic nucleus also influences body temperature, another fundamental circadian rhythm. Core body temperature is not constant throughout the day, but rather follows a predictable pattern, typically reaching its lowest point in the early morning hours, around 4 or 5 a.m., then rising gradually through the day to peak in the late afternoon or early evening. This temperature rhythm persists even in people who remain at complete rest, indicating it is driven by the circadian clock rather than by patterns of activity. Research has revealed that the suprachiasmatic nucleus coordinates temperature rhythms through multiple pathways, affecting metabolic rate, heat generation in brown adipose tissue, and heat loss through changes in skin blood flow and posture. The timing of the temperature minimum is closely linked to the timing of other circadian rhythms, and provides a reliable marker of circadian phase. Perhaps most fascinating is the realization that the suprachiasmatic nucleus not only generates circadian rhythms, but also gates its own sensitivity to light, determining when light exposure can shift the clock and when it has little effect. This phenomenon creates what scientists call a phase response curve, a graph showing how light exposure at different times of day affects the circadian system. Light exposure in the early morning, after the temperature minimum, causes a phase advance, shifting the clock earlier. Light in the evening causes a phase delay, pushing the clock later. But light in the middle of the day, when the circadian system is at its peak, has minimal effect on clock phase. This gating of sensitivity is not controlled by the eyes, but by the suprachiasmatic nucleus itself, which modulates its own responsiveness to the light signals it receives. This self-regulation allows the circadian system to remain stable and resistant to minor fluctuations in light exposure, while still being able to adjust to meaningful changes in the light-dark cycle, such as the changing seasons or travel across time zones. It represents an elegant solution to the challenge of maintaining both stability and flexibility in a changing environment. As you rest here now, in the quiet hours, your suprachiasmatic nucleus is at work, its neurons firing in their characteristic nighttime pattern, low and slow, coordinating the myriad processes that unfold during sleep. It is ensuring that melatonin flows from your pineal gland, that your body temperature drifts to its lowest point, that growth hormone is released, and that cellular repair processes are optimally timed. This tiny structure, these 20,000 neurons, represent the conductor of your biological orchestra, ensuring that every instrument plays its part at precisely the right moment. And as we continue our journey tonight, we will explore the chemical signals this conductor uses, particularly the hormone that serves as darkness's gentle messenger, melatonin. The molecular machinery of time. Deep within each cell of your body, beyond the reach of conscious awareness, a molecular clock is ticking. It is not a clock in any mechanical sense, with gears and springs, but rather an elegant biological system where molecules interact in carefully orchestrated cycles, marking time with remarkable precision. This cellular timekeeper, present not just in neurons of the suprachiasmatic nucleus, but in nearly every cell of your body, represents one of the most fundamental and ancient regulatory systems in biology. The story of how cells keep time begins with genes, those segments of DNA that contain the instructions for making proteins. Among the thousands of genes in the human genome, a subset of approximately 20 genes are classified as core clock genes. These genes do not keep time in isolation, but rather work together in an intricate network of molecular interactions, forming feedback loops that oscillate with a period close to 24 hours. At the heart of this molecular machinery are two positive elements of the clock, proteins called clock and BMAL1. These proteins are transcription factors, meaning their job is to bind to DNA and activate the transcription of other genes. Clock and BMAL1 work together, forming a complex that recognizes specific DNA sequences called E-boxes, short for enhancer boxes, located in the regulatory regions of clock-controlled genes. When the clock BMAL1 complex binds to these E-boxes, it initiates the transcription of two families of genes: the period genes, abbreviated PER, and the cryptochrome genes, abbreviated CRY. In mammals, there are three period genes named PER1, PER2, and PER3, and 2 cryptochrome genes, CRY1 and 2. The transcription of these genes produces messenger RNA, which then travels from the cell nucleus to the cytoplasm, where it is translated into proteins. This is where the negative feedback loop begins. As PER and CRY proteins accumulate in the cytoplasm, they undergo a series of modifications. They are phosphorylated, meaning phosphate groups are added to them by enzymes called kinases. This phosphorylation serves multiple functions. It can affect protein stability, determining how quickly the proteins are degraded, and it can affect protein localization, determining where in the cell the proteins go. The phosphorylation of PER and CRY proteins is not random, but highly regulated. Several kinases have been identified that phosphorylate these clock proteins, including casein kinase 1, epsilon and delta, often abbreviated as CK, 1 epsilon and CK, 1 delta. The rate at which these kinases add phosphate groups helps determine the period of the circadian clock. Mutations that speed up or slow down phosphorylation can shorten or lengthen the circadian period, demonstrating the crucial role of this post-translational modification in timekeeping. Once sufficiently phosphorylated, PER and CRY proteins form complexes with each other and with other regulatory proteins. These complexes then translocate back into the cell nucleus, where they interact with the clock-BMAL1 complex. The interaction is inhibitory. PER and CRY proteins suppress the activity of CLOCK and BMAL1, preventing them from activating the transcription of more PER and CRY genes. This creates the negative feedback that is essential for the clock to oscillate. Early in the cycle, when PER and CRY levels are low, CLOCK and BMAL1 are free to drive transcription. As PER and CRY accumulate, they eventually inhibit CLOCK and BMAL1, shutting down their own production. But PER and CRY proteins are not stable indefinitely. They are continually being degraded by the cell's protein degradation machinery, primarily through the proteasome system. As PER and CRY levels decline due to degradation, the inhibition on CLOCK and BMAL1 is relieved. The transcription factors can once again bind to E-boxes and activate transcription, starting the cycle anew. The entire process, from the initial transcription of PER and CRY genes, through protein accumulation, nuclear entry, feedback inhibition, and eventual degradation, takes approximately 24 hours in mammals. But this core loop does not operate in isolation. There is a second interlocking feedback loop that adds stability and precision to the clock. This auxiliary loop involves genes called REV-ERB alpha and ROR alpha. These genes are also activated by clock and BMAL1, but their proteins have a different function. They regulate the transcription of BMAL1 itself. Reverb. Alpha suppresses BMAL1 transcription, while ROR alpha enhances it. These proteins compete for the same DNA binding sites in the regulatory region of the BMAL1 gene. The balance between REV-ERB alpha and ROR alpha determines how much BMAL1 is produced, and because their own levels are controlled by the core clock BMAL1- per -CRY loop, this creates a second oscillation that reinforces and stabilizes the primary one. This molecular architecture, with its interlocking feedback loops, is remarkably conserved across the animal kingdom. Similar clock gene networks exist in fruit flies, mice, and humans, though with variations in the specific genes involved and their regulation. This conservation could suggest that this mechanism of cellular timekeeping evolved early in the history of complex organisms and has been maintained because of its fundamental importance to survival. Within the suprachiasmatic nucleus, the master circadian pacemaker, these molecular clocks in individual neurons are synchronized with each other through intercellular communication. The neurotransmitter VIV-IP, vasoactive intestinal polypeptide, plays a crucial role in this synchronization. When VIP is released from one neuron and binds to VPAC2 receptors on neighboring neurons, it triggers signaling cascades that can adjust the phase and period of their molecular clocks. This coupling between neurons is what allows the suprachiasmatic nucleus to maintain such robust and precise rhythms. Individual neurons, if isolated, show slightly different periods, some running a bit faster than 24 hours, others a bit slower. But when coupled together through VIP and other signals, they entrain to a common period, oscillating in synchrony like pendulums linked by springs. Outside the suprachiasmatic nucleus, in peripheral tissues like the liver, heart, kidneys, and skin, cells also contain these molecular clocks. For many years, it was thought that these peripheral clocks were simply slaves to the master clock in the brain, passively following signals sent from the SCN. But research has revealed a more complex and interesting picture. Peripheral clocks are not entirely dependent on the suprachiasmatic nucleus for their oscillations. If you take liver cells and culture them in a dish, separated from any neural or hormonal signals from the brain, they continue to show circadian rhythms in gene expression. These rhythms eventually dampen and desynchronize without external cues, but their existence demonstrates that peripheral tissues have intrinsic timekeeping ability. In the intact organism, the suprachiasmatic nucleus coordinates these peripheral clocks through multiple mechanisms. Neural signals, hormonal rhythms, body temperature cycles, and feeding rhythms all serve as time cues that help keep peripheral clocks aligned with the master clock and with each other. This hierarchical organization ensures that the trillions of cellular clocks throughout your body work in concert, timing cellular processes to occur at optimal times of day. The implications of this molecular timekeeping are profound. Clock genes and their protein products do not only regulate their own cyclic production, but also control the expression of thousands of other genes throughout the genome. These clock-controlled genes, as they are called, include genes involved in metabolism, cell division, DNA repair, immune function, and countless other processes. Studies using genome-wide analysis techniques have revealed that approximately 10 to 15% of all genes in a given tissue show circadian rhythms in their expression. Different tissues show rhythms in different genes, reflecting their specialized functions. In the liver, many genes involved in glucose and lipid metabolism are clock-controlled. In the immune system, genes regulating inflammatory responses show circadian variation. In the skin, genes involved in DNA repair and protection from ultraviolet damage peak during the day, when such damage is most likely to occur. This widespread circadian regulation means that the molecular clock is not just a timekeeper, but an organizer of cellular function, ensuring that different processes occur at appropriate times. Metabolism is coordinated with anticipated feeding times. Cell division is timed to avoid periods of high oxidative stress. Immune responses are calibrated to times when pathogen exposure may be most likely, though the precise mechanisms underlying these patterns continue to be explored. The discovery of the molecular basis of circadian rhythms has also revealed why disruptions to the circadian system can have such broad health consequences. When the molecular clocks in cells are misaligned with the external environment, or when different clocks in the body are desynchronized from each other, the temporal coordination of cellular processes breaks down. Metabolic pathways may be active at inappropriate times. DNA repair may not coincide with DNA damage. Hormones may be released when they should be suppressed. This molecular understanding has opened new avenues for therapeutic intervention. Drugs can potentially be designed to target specific components of the clock mechanism, either to adjust clock timing in conditions like delayed sleep phase syndrome or to protect the clock from disruption during shift work. Cancer chronotherapy, the timing of chemotherapy drugs, to coincide with circadian phases that may maximize effectiveness while minimizing toxicity, is showing promise in clinical trials, though more research is needed to establish optimal protocols. As you lie here now in each of your cells, this molecular dance continues. Clock and BMAL1 proteins are binding to DNA, or perhaps being inhibited by PER and CRY. Kinases are adding phosphate groups. Proteins are being synthesized and degraded. The molecular gears are turning, marking time in the darkness, preparing your body for the transition from night to day that will come with the morning light. This invisible cellular clock, ticking away in every tissue and organ, represents one of the most elegant examples of how complex behaviors and physiological processes can emerge from simple molecular interactions. It is a reminder that time itself, at least as your body experiences it, is not merely an external phenomenon, but something generated from within through the patient cycling of molecules in the dark interior of cells. Melatonin, the hormone of darkness. As day transitions to night and the sun dips below the horizon, a remarkable transformation begins to unfold within your body. Deep in the center of your brain, nestled between the two hemispheres, lies a small pine cone-shaped structure called the pineal gland. This gland, once thought by the philosopher Descartes to be the seat of the soul, serves a function both more prosaic and more profound. It produces melatonin, the chemical messenger that signals darkness to the body. The pineal gland is unique among brain structures. While most of the brain is protected behind a barrier that restricts what substances can pass from the bloodstream into neural tissue, the pineal gland is highly vascularized and lacks this barrier. This allows hormones produced by the pineal gland to be secreted directly into the bloodstream, where they can circulate throughout the body and influence distant organs and tissues. The story of melatonin begins with the amino acid tryptophan, which you obtain from dietary protein. Within the pineal gland, tryptophan undergoes a series of biochemical transformations. First, the enzyme tryptophan hydroxylase converts tryptophan to 5-hydroxytryptophan. Then, another enzyme called aromatic amino acid decarboxylase converts this intermediate into serotonin, a neurotransmitter well known for its roles in mood regulation and many other brain functions. During the day, this is where the pathway often ends. Serotonin accumulates in the pineal gland, where it awaits the coming of night. But when darkness falls, a new phase of the pathway is activated. The enzyme arylalkylamine N-acetyltransferase, mercifully abbreviated as AANAT, becomes active and converts serotonin into N-acetylserotonin. Finally, the enzyme hydroxyindole-O methyltransferase, also called ASMT, adds a methyl group to produce melatonin. This final conversion step is the rate-limiting factor in melatonin production. The activity of AANAT in particular shows dramatic circadian and light-dark regulation. During the day, AANAT activity is very low, suppressed by neural signals from the suprachiasmatic nucleus. At night, these inhibitory signals are withdrawn, and AANAT activity increases dramatically, sometimes by 100-fold or more. This creates the sharp day-night difference in melatonin production. The neural pathway controlling melatonin synthesis is complex and multisynaptic, representing the end result of a chain of signals that begins in the suprachiasmatic nucleus. As we explored earlier, the SCN maintains the master circadian rhythm, and one of its primary outputs is the control of this pathway to the pineal gland. During the subjective night, when SCN neurons show low electrical activity, they permit signals to flow through the paraventricular nucleus of the hypothalamus, down through the spinal cord to the intermediolateral cell column, and out to the superior cervical ganglion, a collection of sympathetic neurons in the neck. From the superior cervical ganglion, postganglionic sympathetic fibers travel to the pineal gland. These nerve terminals release norepinephrine, which binds to adrenergic receptors on pineal cells. This binding triggers a cascade of intracellular signals involving the production of cyclic AMP, a common second messenger in cells, which ultimately activates the transcription of the AANAT gene and increases AANAT enzyme activity. The result is a robust circadian rhythm of melatonin secretion. In most people, melatonin levels in the blood remain very low during the day, typically less than 10 picograms per milliliter. As evening approaches and light exposure diminishes, melatonin begins to rise. This increase typically starts 1 to 2 hours before habitual bedtime, in a phase sometimes called dim light melatonin onset, abbreviated as DLMO, which sleep researchers use as a reliable marker of circadian phase. Melatonin levels continue to rise through the early night, reaching a peak sometime between midnight and 4 a.m. in most people, though the exact timing varies with individual chronotype. After this peak, melatonin levels begin to decline, falling to low daytime levels by morning. The entire rhythm forms a bell-shaped curve of secretion across the night, with the duration of elevated melatonin reflecting the length of the dark period. The functions of melatonin are multiple and still being fully understood. Most familiarly, melatonin is involved in sleep regulation, though its role is more subtle than often portrayed. Melatonin does not directly cause sleep in the way that a sleeping pill might. Rather, based on what we understand, it appears to facilitate sleep by promoting physiological and psychological states conducive to sleep onset. It lowers core body temperature slightly. It may reduce alertness. It can promote a sense of drowsiness in some individuals. But perhaps more importantly, melatonin serves as an internal time cue, a signal of darkness that helps coordinate circadian rhythms throughout the body. Many tissues express melatonin receptors, including the suprachiasmatic nucleus itself. When melatonin binds to receptors in the SCN, it provides feedback about the state of darkness. helping to reinforce and stabilize the circadian rhythms generated by the master clock. Peripheral tissues also respond to melatonin. In the cardiovascular system, melatonin may help coordinate the nighttime decrease in blood pressure and heart rate. In the immune system, melatonin receptors on immune cells suggest a role in coordinating immune function with time of day. In various organs, melatonin may help entrain the local peripheral clocks to the central circadian signal. Melatonin also has antioxidant properties, acting as a free radical scavenger. Free radicals are reactive molecules that can damage cellular components, and melatonin can neutralize these molecules, potentially providing cellular protection. Some researchers have proposed that the nighttime rise in melatonin may help protect cells during the vulnerable period of sleep, when repair and restoration processes are occurring, though this remains an active area of investigation. Light exposure, particularly in the evening and night, suppresses melatonin production. This suppression is mediated by the same melanopsin-containing retinal ganglion cells that send light information to the suprachiasmatic nucleus. When these cells detect light, they signal the SCN, which in turn suppresses the neural pathway to the pineal gland, reducing melatonin synthesis. The wavelength of light matters. Blue light, with wavelengths around 460 to 480 nanometers, is particularly effective at suppressing melatonin because melanopsin has its peak sensitivity in this range. This has important implications for modern life. The screens of computers, tablets, and smartphones emit significant amounts of blue light. Evening exposure to these devices can suppress melatonin production, potentially delaying sleep onset and disrupting circadian rhythms. Research has shown that even relatively modest light exposure can affect melatonin. Room lighting that seems dim to our eyes, around 100 lux or so, can suppress melatonin to some degree. Bright indoor lighting of several hundred lux can substantially suppress melatonin. Outdoor light, even on a cloudy day, is far brighter and will completely suppress melatonin production if it reaches the eyes. This sensitivity to light created no problem for most of human history, when the only light available at night came from fire, which is relatively dim and rich in longer wavelengths, rather than the blue wavelengths that most effectively suppress melatonin. But in the modern world, with electric lighting allowing us to create bright environments at any hour, this ancient light sensitivity has become a source of circadian disruption. The use of melatonin as a supplement has become widespread, though its effectiveness varies depending on the context and individual. For certain specific purposes, melatonin supplementation has shown benefits in research studies. For circadian phase shifting, melatonin is most effective when taken at specific times relative to the existing circadian phase. Generally, melatonin taken in the evening, several hours before habitual bedtime, can help advance the circadian rhythm, making sleep onset earlier. Melatonin taken in the morning can delay the rhythm, though this use is less common. The key is timing. Melatonin must be taken at the appropriate phase of the circadian cycle to have the desired shifting effect. For jet lag, melatonin taken at the bedtime of the destination can help accelerate adaptation to the new time zone. Studies have shown modest benefits of melatonin for reducing jet lag symptoms, particularly after eastward travel across multiple time zones. The effectiveness is greatest when combined with appropriate light exposure, which remains the most powerful zeitgeber for the circadian system. As a general sleep aid, melatonin's effectiveness is more variable. In people with normal melatonin production, Taking supplemental melatonin at night may have only modest effects on sleep onset or quality. The benefit appears greatest in individuals who have low endogenous melatonin production, such as some older adults, or in those whose melatonin rhythm is misaligned with their desired sleep time, such as in delayed sleep phase syndrome. The doses used in commercially available melatonin supplements often far exceed physiological levels. While endogenous melatonin production results in blood levels measured in picograms per milliliter, supplements often contain milligram doses that can produce blood levels 100 times higher or more. Whether these pharmacological doses are more effective than lower, more physiological doses remains a matter of ongoing research. Here in the darkness, as you rest, your pineal gland is quietly fulfilling its ancient function. Melatonin molecules are being synthesized from serotonin, secreted into your bloodstream, traveling to tissues throughout your body. They are binding to receptors in your suprachiasmatic nucleus, providing feedback about the state of darkness. They are influencing neurons in your sleep-wake circuits, facilitating the deep rest you are experiencing now. They are scavenging free radicals, protecting your cells. This chemical messenger of night is weaving through your body, coordinating the myriad processes that define the nighttime state, making possible the restoration and renewal that sleep provides. Chronotypes, individual variations in time. If you have ever wondered why some people spring out of bed with the dawn while others struggle to wake before noon, why some are most alert and creative in the early morning, while others don't truly come alive until evening, the answer lies in a fundamental aspect of circadian biology called chronotype. Chronotype is your individual circadian preference, the natural timing of your body's rhythms, and it varies widely from person to person in ways that are both fascinating and consequential. The concept of chronotypes has deep roots in folk wisdom and literature. We have long recognized morning people, often called larks or early birds, who feel most energetic in the early hours and prefer to sleep and wake early. We have also acknowledged evening people called owls or night owls, who reach their peak performance later in the day and prefer to sleep and wake late. Most people fall somewhere between these extremes in the intermediate chronotype category. In formal scientific study, chronotype is typically assessed through questionnaires that ask about preferred sleep and wake times on free days, when work or school obligations do not constrain scheduling. One widely used instrument, called the morningness-eveningness questionnaire, asks a series of questions about sleep preferences, peak times for physical and mental performance, and subjective alertness at different times of day. Studies using such questionnaires have revealed that chronotype is distributed across the population in a roughly bell-shaped curve. The majority of people cluster in the middle, with moderate preferences. About 10 to 20% of individuals fall at the early extreme, qualifying as strong morning types. A similar or slightly larger percentage fall at the late extreme, as strong evening types. The exact percentages vary depending on the population studied and the cutoffs used to define extremes. But chronotype is not merely a matter of preference or habit. Research has revealed that it has a substantial genetic basis. Twin studies, which compare the similarity of traits between identical twins, who share all their genes, and fraternal twins, who share only half, have shown that chronotype is moderately heritable. Estimates suggest that genetic factors account for approximately 40 to 50% of the variation in chronotype between individuals. The search for specific genes that influence chronotype has been fruitful. large genome-wide association studies, which scan the entire genome looking for genetic variants associated with morning or evening preference, have identified hundreds of genetic locations, or loci, that show associations with chronotype. Many of these loci are near or within known clock genes, providing strong evidence that variations in the circadian clock mechanism itself contribute to individual differences in timing. Variations in the period 2 gene, for example, have been associated with extreme morning chronotype, a well-studied mutation that causes familial advanced sleep phase syndrome results in individuals who naturally sleep from approximately 7 or 8 p.m. until 3 or 4 a.m., shifted dramatically earlier than typical. This mutation alters the phosphorylation of the PER2 protein, speeding up the molecular clock and shortening the circadian period, which manifests as an advanced phase of entrainment. Other genetic variants affect different components of the clock mechanism. Variations in the clock gene, the BMAL1 gene, and genes encoding kinases that phosphorylate clock proteins have all been associated with chronotype differences. The cumulative effect of variants across many genes determines an individual's position on the chronotype spectrum. But if chronotype has such a strong genetic component, why does it vary so much? Why would natural selection maintain such a broad distribution of chronotypes in the population? Several hypotheses have been proposed. One compelling idea, sometimes called the sentinel hypothesis, suggests that having a range of chronotypes within a social group may have provided a protective advantage, with some individuals naturally alert early in the night, others in the middle of the night, and still others near dawn. The group could have been less vulnerable to nocturnal threats because someone was likely to be awake and vigilant at any given time. Studies of traditional societies living closer to ancestral conditions have provided some support for this idea. Research on hunter-gatherer groups in Tanzania found that the distribution of chronotypes within camps was such that at least one individual was awake or in light sleep for most of the night. This natural pattern of staggered sleep times could have provided significant survival benefits in environments with predators and other nighttime dangers, though more research is needed to fully establish this connection. Chronotype also changes systematically across the lifespan, following a characteristic developmental trajectory. In early childhood, most children tend toward earlier chronotypes. They wake early and show peak alertness and activity in the morning and afternoon. This pattern serves their developmental needs well, allowing for active play and learning during daylight hours. But as children enter adolescence, a dramatic shift occurs. The circadian clock begins to run later, a phenomenon sometimes called sleep phase delay. Teenagers naturally want to stay up later at night and sleep later in the morning. This is not simply a matter of willful rebellion or poor sleep habits, but rather reflects an underlying biological shift in the circadian system during pubertal development. The magnitude of this shift can be substantial. Studies have shown that adolescent chronotype can be delayed by 1 to 2 hours or more compared to childhood. The deepest point in core body temperature, a reliable marker of circadian phase, occurs progressively later through the teenage years, not reaching its latest point until around age 19 or 20 in males, and slightly earlier, around 17 or 18, in females. This biological delay in adolescent chronotype creates a fundamental mismatch with societal expectations. High schools typically start early, often at 7 or 8 a.m., requiring teenagers to wake at times that are very early from the perspective of their circadian clock. This forces them to curtail sleep, leading to chronic sleep deprivation during a critical period of development. The consequences can include reduced academic performance, increased risk of accidents, mood problems, and various health concerns. Recognition of this biological reality has prompted some school districts to experiment with later school start times for teenagers. Studies of such shifts have generally shown positive results, with students obtaining more sleep, showing improved attendance and academic performance, and reporting better mood and well-being. These findings support the conclusion that the delayed chronotype of adolescence is a genuine biological phenomenon that deserves accommodation rather than resistance. After adolescence, chronotype gradually advances again, shifting earlier through young adulthood and middle age. By their 50s and 60s, many people find themselves naturally waking earlier than they did in their 20s, a shift back toward earlier chronotype. This may reflect changes in the suprachiasmatic nucleus, alterations in sleep homeostasis, reduced exposure to bright light, or a combination of factors. Sex differences in chronotype have also been observed. On average, women tend to have slightly earlier chronotypes than men, particularly before menopause. The magnitude of this difference is not large, perhaps 30 minutes or so in the timing of circadian markers, but it is consistent across studies. After menopause, this sex difference diminishes as women's chronotypes tend to shift slightly later. The modern environment has had a profound effect on chronotype distribution and expression. Artificial light, particularly in the evening, tends to delay circadian rhythms. People with access to electric lighting, computers, and smartphones generally show later chronotypes than those living in environments with only natural light. Studies comparing industrialized and non-industrialized populations have documented these differences clearly. A striking demonstration came from a study where participants went camping in the Rocky Mountains for a week with no access to artificial light or electronic devices. At baseline, the campers showed the typical range of chronotypes, with evening types going to bed and waking late, and morning types on an earlier schedule. But after a week of exposure only to natural light, with no evening light except from campfires, everyone's circadian rhythms shifted earlier, and the spread of chronotypes compressed dramatically. Evening owls advanced their rhythms substantially, becoming much more similar to morning larks. This finding suggests that the modern environment, with evening light exposure and the ability to control our light-dark exposure, has amplified natural chronotype differences and pushed the population as a whole toward later timing. We have, in effect, created an environment that allows evening chronotypes to express their preferences more fully, but also creates greater misalignment between late chronotypes and daytime obligations. The consequences of this chronotype mismatch with social demands have been documented extensively. People with late chronotypes who must wake early for work or school accumulate what researchers call social jet lag, a chronic misalignment between biological time and social time. During the work week, evening types cut sleep short to wake on time, then sleep late on weekends to compensate. This pattern of shifting sleep times is analogous to repeatedly traveling across time zones, hence the term social jet lag. Social jet lag has been associated with various health and performance decrements. Studies have linked greater social jet lag to higher body mass index, increased risk of metabolic syndrome, greater likelihood of depression, and reduced cognitive performance. Based on what we understand, the mechanism appears to involve chronic circadian misalignment, with all the disruptions to metabolism, hormone secretion, and cellular function that such misalignment entails. Evening chronotypes also face specific challenges. Because societal norms favor morning activity, evening types often must force themselves to function at times when their circadian clock is promoting sleep. This can lead to reliance on caffeine and other stimulants during the day, and then difficulty winding down at night despite being tired. Evening types may also engage in behaviors that further delay their rhythms, such as bright light exposure late at night or irregular sleep schedules. However, chronotype is not entirely fixed. While the genetic component is substantial, environmental and behavioral factors can modulate chronotype expression. Light exposure patterns are particularly important. Bright light in the morning can help advance the circadian rhythm, making it easier to wake early. Avoiding bright light in the evening can prevent further delays. Maintaining consistent sleep and wake times, even on weekends, can help stabilize the circadian rhythm. Recent research has also shown that chronotype can shift with behavioral interventions. Studies where evening types were instructed to wake early, get morning light exposure, avoid late-night eating and light exposure, and maintain consistent schedules showed that these individuals could advance their circadian rhythms and improve their daytime functioning. This suggests that while we have genetic predispositions, we are not entirely prisoners of our chronotype. The concept of chronotype raises important questions about how we structure society. Should school start times be adjusted for adolescent chronotype? Should workplaces offer more flexibility in start times to accommodate different chronotypes? Should we recognize that asking a strong evening type to perform optimally at 8 a.m. is comparable to asking a morning type to work at midnight? As you rest here now, your own chronotype is expressing itself in subtle ways. Whether you naturally tend toward early or late timing, whether you are a lark or an owl or somewhere in between, these preferences are rooted in the fundamental neurobiology and genetics of your circadian system. Understanding chronotype helps us appreciate that variations in sleep and wake preferences are not moral failings or choices, but rather reflect genuine biological differences that deserve respect and, when possible, accommodation. When rhythms clash, jet lag, there is a peculiar disorientation that comes with crossing time zones rapidly. You arrive at your destination in daylight, your watch confirming it is morning, yet your body insists it is evening. You feel tired when you should be alert, hungry at unusual times, unable to sleep when night finally comes. This constellation of symptoms, familiar to many travelers, is jet lag, and it represents one of the clearest demonstrations of how powerful and pervasive the circadian system is. Jet lag occurs when there is a mismatch between the timing of the body's internal circadian clock and the external time at the destination. When you travel across 3 or more time zones, particularly in the faster-than-sound manner that air travel allows, your suprachiasmatic nucleus is still entrained to the light-dark cycle of your departure location. It is generating signals appropriate for the time zone you left, not the one you have entered. The symptoms of jet lag arise from this temporal misalignment. At the simplest level, jet lag disrupts sleep. When you attempt to sleep at what is the appropriate bedtime in the new time zone, your circadian clock may be signaling that it is time to be awake. The drive for wakefulness from the circadian system opposes the homeostatic drive for sleep, and the result is difficulty falling asleep and fragmented, unsatisfying sleep when it does occur. Conversely, during the day at your destination, when you need to be alert and functional, your circadian clock may be signaling that it is time to sleep. The circadian nadir, the the point of lowest circadian drive for wakefulness, typically occurs in the early morning hours around 3 to 5 a.m. If you have traveled eastward and this nadir now falls during the afternoon by local time, you will experience profound sleepiness at what should be an active time of day. But jet lag involves more than just sleep disruption. The circadian system regulates a wide array of physiological processes, and when these are misaligned with the local environment and social schedule, myriad problems can result. Appetite and digestion are affected. You may not feel hungry at mealtimes, but experience intense hunger at odd hours. Your gastrointestinal system, with its own circadian rhythms of motility and enzyme secretion, may not be prepared to process food when you eat it, leading to indigestion and changes in bowel patterns. Cognitive performance suffers during jet lag. Attention, memory, and executive functions all show circadian variation, typically performing best during the biological day. When jet lag causes the circadian system to be out of phase with local time, these cognitive functions are impaired. Decision-making becomes more difficult. Reaction times slow. Mistakes become more likely. For business travelers or athletes, these decrements can have significant consequences. Mood and emotional regulation are also affected. People experiencing jet lag often report feeling irritable, less able to regulate their emotions, and sometimes experiencing symptoms of depression or anxiety. This could be related to disruption of the circadian rhythms of neurotransmitters and hormones involved in mood regulation, or simply to the accumulated effects of sleep deprivation and physiological stress. The severity of jet lag symptoms depends on several factors. The number of time zones crossed is obviously important. Travel across 1 or 2 time zones typically causes minimal problems, as this is within the range that the circadian system can accommodate relatively easily. But crossing 3, 4, or more time zones creates substantial misalignment that takes days to resolve. The direction of travel also matters. Eastward travel, which requires advancing the circadian clock to an earlier time, is generally more difficult than westward travel, which requires delaying the clock. This asymmetry reflects a fundamental property of the circadian system. The endogenous period of the human circadian clock is slightly longer than 24 hours, typically running between 24 hours and 15 24 hours, and 30 minutes in the absence of time cues. Because the clock naturally runs slightly longer than a day, it is easier to delay the clock further than to advance it earlier. Westward travel, which effectively lengthens the day, aligns with this natural tendency. Eastward travel, which requires shortening the day, works against it. This is why traveling west coast to east coast in the United States, crossing 3 time zones eastward, often feels harder than the return journey westward. Individual factors also influence jet lag severity. People with more rigid circadian systems, who maintain very regular schedules, may find it harder to adjust to new time zones. Younger individuals generally adapt more quickly than older adults, perhaps reflecting greater flexibility in their circadian systems. Chronotype may play a role as well. Evening types, whose circadian systems are more naturally delayed, may find eastward travel particularly challenging, while morning types may struggle more with westward travel. The process of recovering from jet lag, known as re-entrainment, occurs gradually as the circadian clock adjusts to the new time zone. The suprachiasmatic nucleus must shift its phase to align with the light-dark cycle at the destination. This shift occurs at a rate of roughly 1 to 2 hours per day for most people, though the rate varies between individuals and depends on the direction of travel. Light exposure is the most powerful tool for accelerating re-entrainment. Properly timed light exposure can help shift the circadian clock in the desired direction. For eastward travel, where you need to advance the clock earlier, bright light exposure in the morning at the destination is beneficial. It signals the suprachiasmatic nucleus that this is the beginning of the day, helping to phase advance the clock. For westward travel, where you need to delay the clock later, evening light at the destination helps. It extends the perceived day, encouraging the clock to delay its phase. However, there is a complication. The phase response curve, which describes how light affects the clock at different times, shows that light in the early part of the biological night causes delays, while light in the later part of the biological night causes advances. This means that if you are severely jet-lagged, light at the wrong time could shift your clock in the wrong direction, making matters worse. For example, if you have traveled eastward and your biological clock still thinks it is evening, when it is actually morning at your destination, morning light might fall during what your clock perceives as late biological night, potentially causing a phase delay instead of the desired advance. Some travelers use melatonin supplementation to help with jet lag. The logic is that melatonin, taken at the appropriate time, can help shift the circadian clock. For eastward travel, melatonin taken in the evening at the destination, at what would be earlier than your usual bedtime by your home clock, may help advance the circadian rhythm. For westward travel, melatonin taken in the early morning might help delay the rhythm, though this is less commonly practiced. Research on melatonin for jet lag has shown modest benefits, particularly for eastward travel across multiple time zones. However, the effects are not dramatic, and melatonin works best when combined with appropriate light exposure and behavioral strategies. The timing of melatonin administration is crucial, as taking it at the wrong time could theoretically shift the clock in the undesired direction. Behavioral strategies can also help minimize jet lag. Before travel, some people begin adjusting their sleep schedule toward the destination time zone. Going to bed and waking up an hour earlier each day for a few days before eastward travel, for instance, can pre-adapt the circadian clock and reduce the magnitude of the shift needed upon arrival. Upon arrival, immediately adopting the local schedule for sleep, meals, and activities helps entrain the circadian system to the new time zone. Avoiding naps, as tempting as they may be, helps consolidate sleep at the appropriate time. If you must nap, keeping it brief, no more than 20 to 30 minutes, and earlier in the day can provide rest without significantly disrupting nighttime sleep. Staying hydrated during travel is important, as dehydration can exacerbate feelings of fatigue. Avoiding excessive alcohol and caffeine, both of which can disrupt sleep, helps maintain sleep quality. Some travelers use strategic caffeine consumption to help maintain alertness during the day at the destination, being careful to avoid it in the hours before bedtime. For frequent travelers who cross multiple time zones regularly, some choose not to adjust to the destination time zone at all for short trips. Business travelers who will be at a destination for only a day or two might maintain their home schedule, conducting meetings at times that align with their circadian rhythm, even if these are unusual hours locally. This avoids the disruption of re-entrainment for a short visit, though it limits the ability to participate in local evening or early morning activities. For longer stays, full re-entrainment is generally preferable, as maintaining home time becomes increasingly difficult and socially isolating. The body will naturally begin to entrain to the local light-dark cycle regardless of intentions, and attempting to resist this process can create chronic circadian misalignment. Athletes traveling for competitions face particular challenges with jet lag, as even small decrements in performance can be meaningful. Sports science research has developed protocols to minimize jet lag effects, often involving traveling well in advance of competitions to allow time for full re-entrainment, strategic light exposure and melatonin use, and careful scheduling of training sessions to align with circadian rhythms. As you rest here now, in the quiet stillness of night, your body is where it should be in time as well as space. Your circadian clock is aligned with the world around you. The darkness outside matches the darkness your body expects. For those who have recently crossed time zones, this alignment is disrupted, and their bodies are still adjusting, still searching for synchronization with the new environment. It is a reminder that we are deeply temporal beings, not easily displaced from our place in the daily cycle, and the challenges of shift work. If jet lag represents a temporary disruption of circadian rhythms, shift work represents something far more concerning: chronic, ongoing circadian misalignment. Around the world, millions of people work during hours when their circadian biology is programmed for sleep. They are nurses and doctors staffing hospitals through the night, factory workers maintaining production lines, police officers and firefighters keeping communities safe, truck drivers transporting goods. The modern 24-hour economy depends on their labor, but this work comes at a significant biological cost. Shift work, particularly night shift work, creates a fundamental conflict between the body's internal clock and the external demands of work schedules. Unlike jet lag, where the circadian system can gradually re-entrain to a new stable time zone, shift workers often cannot fully adapt because their schedule changes frequently, or because they are exposed to daylight during their commute, or because they revert to a normal day-oriented schedule on their days off. The result is chronic circadian disruption. The suprachiasmatic nucleus continues to generate signals appropriate for a day-active schedule, even as the person tries to sleep during the day and work through the night. Melatonin rises in the evening and falls in the morning, regardless of when the person is trying to sleep. Core body temperature follows its usual pattern, reaching its nadir in the early morning hours, when the shift worker is trying to stay alert and perform demanding tasks. This misalignment has immediate effects on alertness and performance. The circadian drive for wakefulness is lowest in the early morning hours, typically between 3 and 5 a.m. A night shift worker at this time is fighting against biology. Even with adequate prior sleep, the circadian system is promoting sleepiness, making it difficult to maintain attention, respond quickly, and make good decisions. The risk of errors and accidents is substantially elevated during night shifts. Studies across various industries have documented this increased risk. Medical errors in hospitals show a circadian pattern, with more errors occurring during night shifts. Industrial accidents peak in the early morning hours. Truck drivers have more accidents during night driving. The Chernobyl nuclear disaster, the 3 Mile Island incident, and the Exxon Valdez oil spill all occurred during night shifts, when circadian impaired performance may have contributed to the catastrophes. Beyond acute performance effects, chronic shift work has been associated with numerous health problems. Shift workers have higher rates of cardiovascular disease, including hypertension, myocardial infarction, and stroke. The mechanisms are not entirely clear, but may involve chronic disruption of blood pressure rhythms, inflammation, and metabolic dysfunction. Metabolic health is significantly affected by shift work. Shift workers have higher rates of obesity, metabolic syndrome, and type 2 diabetes. The circadian system normally coordinates metabolism with anticipated feeding times, preparing the body to process nutrients when food is expected. When eating occurs during the biological night, when metabolic efficiency is low and the body is not prepared for food intake, this can lead to metabolic dysfunction. Eating during night shifts, when insulin sensitivity is naturally lower and metabolic processes are down-regulated, may contribute to weight gain and metabolic problems. Studies have shown that the same meal consumed at night produces higher glucose and insulin responses than when consumed during the day, suggesting that the timing of eating relative to circadian phase affects how nutrients are metabolized. Gastrointestinal problems are common among shift workers. Irregular meal times, eating during the biological night, and disrupted sleep all contribute to digestive issues. Shift workers report higher rates of peptic ulcer disease, gastroesophageal reflux, and irritable bowel syndrome. The gastrointestinal tract has its own circadian rhythms of motility, enzyme secretion, and barrier function, and disrupting these rhythms has consequences for digestive health. Cancer risk may also be elevated among shift workers. The International Agency for Research on Cancer has classified shift work involving circadian disruption as probably carcinogenic to humans. Epidemiological studies have found increased rates of breast cancer among female night shift workers and elevated rates of prostate cancer among male shift workers. The magnitude of the increased risk is modest but consistent across studies. The mechanisms that may link shift work to cancer are multifaceted and not yet fully understood. Disruption of circadian rhythms appears to affect cell cycle regulation, DNA repair, and apoptosis, the programmed death of damaged cells. Light exposure at night suppresses melatonin, and melatonin has been shown to have potential anti-cancer properties in laboratory studies. Disrupted sleep and circadian rhythms may also affect immune surveillance of cancer cells. While the evidence suggests a connection, much remains to be understood, and researchers continue to investigate these complex relationships. Mental health and well-being are also affected by shift work. Depression, anxiety, and burnout are more common among shift workers. The chronic sleep deprivation, circadian disruption, and social isolation that often accompany shift work all contribute to mental health challenges. Relationships and family life can suffer when one partner is on a schedule that is out of sync with the rest of the household. Social connections become harder to maintain when work hours fall during times when friends and family are free. Despite these challenges, shift work remains a necessity in many industries. The question then becomes, how can we minimize the harm? Research has identified several strategies that may help. One approach is to design shift schedules that work with, rather than against, circadian biology. Forward-rotating shifts, which move from day to evening to night shifts rather than backward, are generally better tolerated because they align with the circadian system's natural tendency to delay rather than advance. Limiting the number of consecutive night shifts can reduce cumulative sleep debt and circadian disruption. Some research suggests that no more than 3 to 5 consecutive night shifts should be worked before a period of recovery. Adequate time off between shifts to allow for recovery sleep is crucial. Rapidly rotating shifts that change every 1 to 2 days may allow the circadian system to remain on a day-oriented schedule, though at the cost of perpetual circadian misalignment during night work. Light exposure can be used strategically to help shift the circadian clock. Bright light exposure during the night shift can help maintain alertness and if sustained over multiple nights, can promote some degree of circadian adaptation. However, this must be balanced against the need to avoid light when trying to sleep during the day. Wearing dark sunglasses when commuting home after a night shift can prevent morning light from shifting the clock in the wrong direction. Napping before or during night shifts can help reduce sleep debt and improve alertness. A nap of 1 to 2 hours before starting a night shift can boost performance. Short naps during night shift breaks can help maintain alertness, though care must be taken to allow time for sleep inertia, the grogginess immediately after waking, to dissipate before returning to critical tasks. Caffeine, used judiciously, can help maintain alertness during night shifts. However, caffeine has a half-life of several hours, meaning it remains in the system for a long time after consumption. Consuming caffeine late in a night shift can interfere with subsequent daytime sleep. Strategic caffeine use, consuming it early in the shift and avoiding it in the last few hours, may provide benefits without disrupting sleep. Creating an optimal sleep environment for daytime sleep is essential. This means darkening the bedroom completely, using blackout curtains or eye masks, reducing noise through the use of earplugs or white noise machines, keeping the bedroom cool, communicating with family members about the importance of minimizing disruptions during daytime sleep hours. These may seem like small measures, but for shift workers they can make the difference between obtaining adequate sleep and not. From a societal perspective, there is growing recognition that shift work schedules should be designed with worker health and well-being in mind, not only operational efficiency. Some European countries have implemented regulations limiting night shift work, providing health screenings for shift workers, and offering compensation for the health risks of non-standard schedules. As evidence of the health impacts of shift work continues to accumulate, pressure may grow for similar protections in other regions. For the millions of people who work shifts out of necessity, understanding the biological challenges they face is important. Shift work is not simply a matter of adjusting one's schedule. It represents a fundamental conflict between biological time and social time, between ancient circadian rhythms and modern economic demands. The toll it takes in terms of health, well-being, and quality of life is substantial, even if often invisible to those who work traditional daytime schedules. As you rest here tonight, aligned with your circadian rhythm, sleeping at a time when your body expects sleep, you are experiencing a luxury that shift workers often cannot. The darkness outside matches the darkness signals in your brain. Your body temperature is low. Your melatonin is high. Everything is coordinated, synchronized, as it should be. For those working through the night at this moment, these harmonies are disrupted, and their bodies pay the price, living in harmony with your clock. After our journey through the intricate machinery of the circadian system, from the molecular clockwork in each cell to the master pacemaker in the brain, from the chemical messengers that signal time to the consequences when these rhythms are disrupted, we arrive at a gentler place, a consideration of how to live well within the constraints and gifts of your biological clock. The fundamental principle is simple to state, though sometimes challenging to implement. Respect your circadian rhythm. Recognize that your body is not a machine that can perform equally well at any hour. It is a biological system optimized by millions of years of evolution to function in coordination with the rotation of the Earth, the cycle of light and darkness, and the regular alternation of activity and rest. Light is your most powerful tool for maintaining a healthy circadian rhythm. Expose yourself to bright light, particularly sunlight, early in the day. Morning light helps anchor your circadian system to the day-night cycle, strengthening all the rhythms that cascade from this central timing signal. A walk outside within an hour or two of waking, even on cloudy days, provides light exposure that is far brighter than most indoor lighting. This morning, light helps you feel alert during the day and promotes better sleep at night by reinforcing the appropriate phase of your circadian clock. Conversely, as evening approaches, begin to dim the lights in your environment. Our modern world bathes us in light long after sunset, which can delay the circadian clock and suppress melatonin production. Reducing light exposure in the hours before bed helps your circadian system recognize that night has arrived. If you must use electronic devices in the evening, consider using apps or settings that reduce blue light emission, as blue wavelengths are particularly effective at suppressing melatonin and alerting the brain. Consistency in your sleep-wake schedule is remarkably important. Going to bed and waking up at roughly the same time each day, even on weekends, helps keep your circadian clock stable and properly entrained. When sleep times vary widely from day to day, the circadian system struggles to maintain a clear phase, and you may experience perpetual mild jet lag even without traveling. This is the social jet lag we discussed earlier, and minimizing it by keeping a more regular schedule can improve sleep quality, daytime alertness, and long-term health. This does not mean you must adhere to a rigid schedule with no flexibility. Occasional variations are fine and normal, but large variations, sleeping 4 hours later on weekends than on weekdays, for instance, create circadian disruption. Aim for consistency within an hour or so across different days of the week. Your body will thank you with more stable energy, better sleep, and improved overall well-being. Pay attention to meal timing as well. The circadian system coordinates metabolism with anticipated feeding times, and eating in alignment with your circadian rhythm supports metabolic health. Breakfast in the morning, when metabolism is primed for food processing, and minimizing late-night eating, when metabolic efficiency is low, can support both circadian rhythms and metabolic health. Some research suggests that time-restricted eating, consuming all food within a consistent window of 8 to 12 hours per day, aligned with daytime hours, may have metabolic benefits, though more research is needed to establish optimal practices. Exercise and physical activity also interact with the circadian system. Regular exercise tends to strengthen circadian rhythms and can help improve sleep. The timing of exercise may matter. For many people, exercising earlier in the day or in the late afternoon is ideal, as it can help strengthen circadian amplitude without disrupting evening wind down. Intense exercise too close to bedtime may be alerting and interfere with sleep in some individuals, though this varies from person to person. If you are an evening chronotype, a night owl struggling in a morning lark world, there are strategies that may help. Morning light exposure is particularly important for evening types, as it can help advance the circadian clock earlier. Avoiding bright light late at night is crucial. Keeping a consistent sleep schedule, even on weekends, helps prevent the clock from drifting even later. Some evening types find that gradually shifting their bedtime earlier by 15 to 30 minutes every few days can help them adjust to earlier schedules over time. If you are a morning type living with evening types, perhaps in a household where different family members have different chronotypes, understanding and respecting these differences can reduce conflict. Recognize that when an evening type teenager wants to sleep until noon on weekends, this is not laziness, but biology. When a morning type partner is alert and energetic at 7 a.m., while you are barely conscious. This is not a judgment on your character, but a reflection of different circadian wiring. For those who must occasionally disrupt their circadian rhythms, whether for travel or other reasons, planning can help minimize the impact. Before long-distance eastward travel, gradually shifting sleep earlier for a few days can pre-adapt the circadian clock. Upon arrival, immediate exposure to morning light at the destination helps re-entrainment. Avoiding naps on the day of arrival, as tempting as they may be, helps consolidate sleep at the appropriate time in the new time zone that first night. If you work shifts or have irregular schedules, the advice becomes more complex because you are facing a fundamental conflict between your biology and your obligations. Focus on sleep quality when you can sleep. Create the best possible sleep environment. Use light strategically to help maintain alertness when needed. Prioritize sleep and recognize it as a non-negotiable biological necessity, not a luxury. Seek schedule consistency when possible and advocate for shift schedules that minimize circadian disruption. Throughout all of this, cultivate an awareness of your own circadian rhythms. Notice when you naturally feel most alert, when you feel sleepy, when you perform best mentally and physically. This self-knowledge allows you to plan important tasks for times when your circadian system supports high performance. Schedule demanding cognitive work for your circadian peak, typically late morning or early afternoon for most people. Save routine tasks for times when alertness naturally dips, like early afternoon. Recognize that your relationship with your circadian rhythm changes across your lifespan. The sleep patterns that served you well as a young adult may not be optimal as you age. Adolescents need more sleep and naturally have later chronotypes than adults. Older adults may find they wake earlier and need less total sleep, though the quality of sleep may decline. These changes are normal and expected. Adjusting your expectations and habits to match your current circadian biology, rather than fighting against it, promotes better outcomes. Perhaps most importantly, grant yourself compassion. You live in a society that often demands things of you that conflict with your circadian biology. You may have work or family obligations that require you to wake earlier than your body prefers, to stay up later than is ideal, to maintain schedules that create chronic circadian disruption. These conflicts are real, and their effects on your well-being are not imaginary or a sign of weakness. The circadian system is ancient, forged over billions of years of evolution in organisms living on a rotating planet. The modern world, with its electric lights and global travel and 24-hour economy, is new, a blink in evolutionary time. Your biology has not had time to adapt to these novel demands. When there is conflict between your circadian rhythm and the demands of modern life, the fault does not lie with you or your supposed inability to adapt. The fault lies with a mismatch between ancient biology and contemporary society. You cannot always change your circumstances, but you can make informed choices within the constraints you face. You can prioritize sleep when possible. You can seek light at appropriate times. You can advocate for schedule flexibility. You can choose to honor your body's need for regular rhythms rather than treating sleep as an inconvenience to be minimized. In doing so, you acknowledge a profound truth. You're a biological being, rhythmic and temporal, connected to the rotation of the earth and the cycle of day and night. This is not a limitation, but a fundamental aspect of what you are. Your body keeps time. It has done so since the first cells evolved circadian clocks billions of years ago. and it will continue to keep time, marking the hours, coordinating the myriad processes that sustain your life as long as you live. Respect this timekeeper. Honor it. Work with it rather than against it. And in doing so, you may find that life becomes a little easier, sleep a little deeper, days a little brighter, and the rhythm of existence a little more harmonious with the ancient music of biological time. Closing. As we come to the end of our journey through the science of circadian rhythms, the story we have told is one of exquisite biological precision, of molecular clocks ticking in each cell, of a master pacemaker orchestrating the body's many rhythms, of light and darkness guiding these ancient cycles, and of what happens when these rhythms are disrupted by modern life. You have traveled from the first observations of plants tracking the sun to the discovery of clock genes, from the tiny suprachiasmatic nucleus to the sprawling network of clocks throughout your body, from the chemistry of melatonin to the genetics of chronotype, from the disorientation of jet lag to the toll of shift work. And throughout, the central theme has remained. Time is not merely something external that we measure with clocks and calendars. Time is something your body generates, a fundamental property of living systems. The circadian system, with its 24-hour oscillations, connects you to the cosmos in a direct and tangible way. The rotation of the Earth, which creates the cycle of day and night, has shaped your biology so deeply that you carry within you a representation of this rotation, a model of solar time that continues to run even in constant conditions. You are, in a very real sense, a temporal creature, existing not just in space but in time, with your physiology and behavior organized around the fundamental rhythm of the spinning planet. Understanding this can change how you live. It can help you appreciate why sleep matters, why consistency in your schedule supports well-being, why fighting against your circadian rhythm creates difficulty. It can help you be more compassionate with yourself when you struggle with early mornings or late nights, recognizing that these struggles reflect genuine biological realities, not personal failings. It can also help you be more compassionate with others whose circadian rhythms differ from yours, whether that is a teenager who cannot wake easily at dawn, a partner whose chronotype is dramatically different from your own, or a shift worker whose health suffers from chronic circadian disruption. The story of circadian rhythms is still being written. Researchers continue to uncover new aspects of how the clock works, new genes involved in timekeeping, new consequences of circadian disruption, new strategies for supporting healthy rhythms. The 2017 Nobel Prize for the molecular mechanisms of circadian rhythms marked a milestone, but it was not an ending. The field continues to grow, to reveal new layers of complexity and new possibilities for intervention. For now, in these quiet hours, your circadian system is doing its work. The molecular clocks in your cells are ticking through their cycles. Your suprachiasmatic nucleus is generating its characteristic nighttime pattern of activity. Melatonin flows through your bloodstream. Your body temperature has reached its minimum. Your cells are engaged in repair and restoration. All of this happens without conscious effort or awareness, the gift of billions of years of evolution, the inheritance of every organism that has lived on this rotating world. As you drift towards sleep, or simply rest in this state of peaceful relaxation, you can take comfort in knowing that you are part of an ancient rhythm, connected to all living things that mark time, from the simplest bacteria to the most complex mammals. The circadian system is a reminder that we are not separate from nature, but deeply embedded in it, shaped by it, synchronized to it. The night will eventually give way to day. Your circadian clock will sense the coming dawn even before you wake. Your body will prepare for the transition to wakefulness, raising your temperature, suppressing melatonin, coordinating the shift from night to day. And when you wake, you will begin a new cycle, another turn of the great wheel of circadian time. If you enjoyed this exploration of circadian rhythms, please like and subscribe for more journeys into the science of sleep and the mysteries of the mind. Leave a comment below sharing your own experiences with circadian rhythms, your chronotype, or how understanding this biology has affected your life. For now though, simply rest. Let the knowledge of how your body keeps time settle gently in your mind. Let the appreciation of this ancient, elegant system wash over you. Let yourself be cradled by the rhythm that has guided life for eons, the gentle pulse of circadian time. Sleep well and good night.